While the awesome research opportunities you can get if you work in neurosurgery aren't the only reason you should go into neurosurgery, they are a very cool added bonus. So just going to talk a little bit about just one figure from my recent paper in Nature Neuroscience and what I think is cool about this figure and why I do things this way. The way I think about a figure one in a paper is that I want this figure to be an introduction to what the reader is going to be learning about in this study. So whether it's a summary of techniques or an introduction to the kind of questions or data that you're going to be seeing throughout, it should give the reader an idea of what's to come. So what I've done in this figure is on the top, you can see I have a general experimental timeline where I show that, we're using this samples from this part of the brain, the hippocampus, surgically resected, and then we are slicing them into very thin slices on the day of surgery, then plating them, culturing them for a few days before doing the final experiment day where we plate them on the high-density microelectroarrays to do the electrophysiologic recordings. On the next line below, I show an example of a stained hippocampal slice from which we recorded, and then highlight the area of the hippocampal slice that's on the high-density MEA, and then next to that, you show just a sample of the raw data showing activity in one part of the hippocampal slice. So that's, where we're getting our data from. I think this is a way to kind of show the reader, okay, on top, this is generally the paradigm of how we're getting our data. And then the next row, I'm showing, and this is literally what you're going to be looking at. When you're looking at the later figures, you're going to be looking at data from hippocampal slices, where this is kind of the type of area that we get a recording from, and this is the kind of raw data that we see as we're doing the experiment. And now on the final part at the panel at the bottom of this figure, I show a little glimpse of what's to come. Namely, first I'm showing that we transduced these slices with an adenoviral vector, an AEV vector, to get them to express a channel rhodopsin, and in this case, HCKCR1. It's an inhibitory channel rhodopsin, and I'm showing the general expression that we see throughout the hippocampus in one of these slices, the same one that's shown above, and then with some zoom-ins of the neurons that are expressing this staining, showing it looks like there's multiple types of neurons that are doing this, both in the granule cell layer as well as in the outer layers of the hippocampus. And then finally, all the way at the screen bottom right corner, I show an example of provoked epileptiform activity, where we see coordinated activity, bursting activity happening, and showing it both in a kind of a power heat map, as well as just the raw raster plot of all these individual units firing. So the reason I did figure on this way is so that we could, again, introduce the reader to what we're doing in general, where is this data coming from, and what is the kind of data you're going to be seeing in the rest of this paper. It's kind of a grounding, orienting, and giving you a little taste of, if you're interested in this, keep reading the paper.